Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After our speaker is done with the presentation, I will ask your questions to our speaker. Your questions will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Eric Isberg from Savillex.
Thank you, Leah. Hello, everyone, and thank you for attending today's presentation. Our agenda will begin with an introduction to Savillex and our fluoropolymer container technology. I will then discuss key attributes of biocontainer design for cell therapy applications. After this, I will describe a case study of the use of container closure integrity testing for bottles used for a cell therapy application. We will then end with a Q&A session. So let's get started. For the bioprocess market, Savillex is focused on PFA, FEP, and ETFE fluoropolymer bottles and vials. We have two adjacent manufacturing facilities here in Eden Prairie, Minnesota. One is dedicated exclusively to our ready-to-use product line. We were the first company to stretch blow mold fluoropolymer bottles, and we are the only supplier of ready-to-use fluoropolymer bottles and vials. Fluoropolymers have several advantages over standard bottle products. Extremely high purity, very low extractables, thermal stability in a very wide temperature range, and unmatched durability and essentially unbreakable during use. Now, cell therapies require container integrity to protect products from contamination. And part of that is closure torque. Closure torque is key to ensuring this container integrity. At the same time, containers must be usable in a clinical setting where the use of multiple glove layers is very common. Now, the problem with standard containers for cell therapy applications is a danger of very high extractable levels. Also, the closure seals used in these laboratory bottles are really not designed for critical applications. The closures themselves are commonly made from a different material from the bottle. And as you can see in the picture in the lower right-hand corner, this is what can happen when the closure and the bottle are made from different materials. You can actually have the closure fail, whereas the bottle does not. Also, some laboratory bottles use insert seals within the closure system, and those are used usually to compensate for a very poor closure design. Now, the Purelex bottle that we manufacture has a superior closure design. It ensures a much better seal and a better protection of the contents. There's a single product contact material in both the bottle and the closure, and a very high precision molded sealing surfaces. We also use a ferrule style seal, which works even at liquid nitrogen temperatures. And an illustration of that seal can be seen at the bottom of this slide. Now, I talked a little bit about closure torque. Superior closure design allows better and more precise closure torque settings. And a validation of torque is a very important step for critical applications. You know, whether it's lower torques for general use, mid-level torques for long-term storage applications, or even higher or custom set torques for cryogenic applications. Our case study today is an autologous tissue graft. The product is stored and shipped at 2 to 8 degrees in our 100 ml bottles. Although they successfully tested the Savillex bottles during an actual product shipment to their clinical sites, Some concerns remained about the use of, or the ease, of removal of the closures, again, at the surgical suite. So a decision was made by the customer to perform application-specific validation of the closure torque using CCIT methods. The goal was to determine the lowest acceptable torque for their process. A study was performed to quantitatively measure the associated leak rate for the container closure system using helium mass spectrometry. The leak test method was performed using the standard helium test method ASTM F2391. The bottles were prepared at 5 different torque specifications in an attempt to determine the optimal torque for the application. At the same time, control containers were torqued to an identical torque specifications, but included a laser drill defect, 0.2 micron in nominal diameter. As part of the method, a feasibility study was performed to confirm the fitness of helium leak technology for the assessment of the bottle closure system and to determine the optimal test time. The 20-second time point was chosen for the leak test, as it was at least 1 full order of magnitude between the negative and positive control helium leak rates. And there's some more detail at the bottom of the slide if you're interested in the Kirsch criterion and the justification for using that for this study. This slide gives an overview of the results of the test method. The results of the study indicated that the PureLex bottle torqued to at least 17.5 inch-pounds yielded helium leak rates below the acceptance limit and therefore passed, confirming the integrity of the bottle closure package system at this torque or higher. So as you can see from the summary, at 17.5 inch-pounds, all 10 bottles passed and the control failed, indicating that the defect, the laser drill defect, was actually detected during the testing, as it should be. So hopefully this study showed that superior closure design in our PureLex bottles ensures a better seal, allows more precise torque settings, and yields better protection of the contents. Validation of closure torque is a very important step for critical applications, particularly in cell therapy and regen medicine. Savillex offers application-specific testing to support for your critical processes. And that's the end of my presentation. I'd like to thank you for your time. For more information about our products, please feel free to visit our website. We will be preparing a technical note based on this presentation, which should be available very soon.
Are you ready for questions, Eric?
Yes, I am.
Okay, so the first question is, why are different materials used for bottles and bottle closures?
That's a good question. There are usually a couple of reasons why that occurs with bottle systems. Sometimes different methods are used to manufacture the bottle and the closure. So commonly the closures are injection molded, but the bottle itself could be extrusion molded or blow molded. Another reason is that some materials really are not good at creating adequate seals. So sometimes the bottle material is not a good material, so then they use a closure that's made out of a better sealing material. And another reason why different materials are used is that galling can occur. So if you have a bottle and a closure made out of the same material and it's not a fluoropolymer, sometimes that can gall during use. And what the galling will do, it makes it difficult to remove the closure, but also it can create particles because of those surfaces rubbing together. In our case, again, with the fluoropolymers, they're self-lubricating materials, so you don't get any galling and you don't get any particle generation. I hope that answers the question.
That I think so. And then, when should CCIT be used to validate the processes using bottles?
I think it's really up to the end user, but we recommend it whenever you're storing a finished product. And the reason why we used cell therapy and regen medicine as our case study today is that is considered a final product. It's regulated by device regulations. And so this particular customer is very interested in doing container closure integrity testing for their process. But, I think it could be used whenever, absolute container closure integrity is required for your process.
Okay, well, speaking of that, we have a question. I'm assuming the leak testing was performed at ambient temperatures. Did you generate any data for CCI at liquid nitrogen temperatures?
We did not in this study because this was a refrigerated product. They did perform it at room temperature in a general laboratory setting. We at Savillex have created our own data set at liquid nitrogen temperatures, but it was a different study. It wasn't looking at helium as a tracer. We were just doing pressure decay testing. And we do have that technical report available at our website, or you can contact me and I can provide that to you.
Okay, and another question. Do you think the differences in bottle material or form could affect evaporation rates of the chemicals inside?
Oh, I'm sorry. Can you repeat that question?
Yeah. So do you think that the differences in bottle material or form could affect evaporation rates of the chemicals inside?
Yes, absolutely. Certain materials are much a better barrier against moisture loss. Fluoropolymers are, excuse me, an excellent barrier depending on the material. But some other materials, polycarbonate, PETG, also have a different vapor transfer rate. So I think you really need to understand that for your process, whether you're using a solvent system, whether you're using an aqueous solution, you're going to have a different tendency for vapor loss to occur over time. So we offered that testing. We will happily do that. So if you have a particular solvent, for example, we can certainly test that in our laboratory to show the difference between different bottle materials. One other thing to add to that is the container closure system is very critical to that as well, because you will get the moisture transmission through the material, but you could also get it, through the closure system. So having a really tight closure system, a very secure one, is very important.
Okay, great. Well, thank you, Eric. It looks like those are all the questions we have in the webcast. If you can have any more questions, you can continue to submit them and we'll pass them along to Eric and he can follow up with you directly. But for the webcast, we want to thank you for joining us. The recorded version will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.